Yeah, I recently graduated from Biostat in Michigan. So today I'm going to talk to you about my PhD, one of my PhD work on a segmentation-free factor analysis tool for sub-micron dilution spatial transcriptomics data. So spatial technologies have two major factors to consider. First is the spatial resolution here on the x-axis, and the number of genes profile. So imaging, there are two major types. Imaging-based technologies, they can measure the precise location of every single transcript. But the downside is you can only measure a pre-designed gene panel. And also, until recently, it has been limited by its throughput. So that's both the number of gene profile and area of the view. Sequencing-based technology, on the other hand, can almost unbiasedly profile the whole transcriptome. But earlier technologies have been of relatively low resolution at multicellular level. Recently, technologies like 6-scope and TheraSeq have achieved sub-micron resolution that's comparable to microscope. So our method is designed for any subcellular resolution spatial transcriptomics. So I will take 6-scope as example. 6-scope is developed here in Michigan. It's a sequencing-based, next generation sequencing-based technology. So it repurposes the lunar sequencing machine. Here, where the surface of array with attached barcode. So each of the reads cluster will share a single unique spatial barcode, and we'll call that a pixel. And then the frozen tissue section is placed on this physical array, and the RNA is captured down to this array. So when we sequence the captured molecule, we recover both the cDNA and the spatial barcode at the same time. So the 6-scope was initially used MySeq, later HiSeq, and now using NovaSeq, with increased spatial area and increased the transcript density. So currently, the typical resolution is below 1 micrometer, and the transcript density is from 1 to over 20 molecules per square micron. However, this high resolution data does not automatically enable high resolution inference. So on the left is what we observe from the data, the spatially resolved transcript. So in the end, the data you get is a table with pixel ID, X and Y axis, the transcript ID, and the number of copies. So the data is quite sparse, that each pixel can contain only a few molecules. But the first thing we want to do inference is to get individual cells and their cell type and functional identity. So the current state-of-art approach is to use cell segmentation. Cell segmentation relies on nuclear staining and cell boundary markers. But those both type of information is often unavailable, especially for sequencing-based technologies. So even when it is feasible, cell segmentation is still very challenging. Because fundamentally, the 2D capture of the 3D tissue section is incomplete. And cells and nuclei are often only partially captured by this 2D view. And cells do have irregular cell shape. So, for example, the nuclei of some cells may only be partially captured, or in this case, completely missed. So those cells will be missed from cell segmentation. And also some cells do have multiple nuclei, like muscle cells or hepatocytes. An alternative approach is to use a fixed grid-based low resolution analysis. So here we can use all the transcript in the data. But the downside is it loses the original high resolution provided by the technology. And also each of such grid will often contain multiple different cell types, and your algorithm should be able to deal with that. The ideal solution, however, is to get high resolution to get the cell type annotation at each single pixel, and hopefully do so efficiently for a large field of view. And this is what we want to achieve. Yeah, so this is the intuition of how FICTURE works. We start from the grid-based approach that we take aggregate individual pixels into hexagons, and for each of the hexagons, we infer the mixture probability over the different cell types. Here are the factors. Those factors could be learned from the data, or we could also initialize with prior knowledge, or we can use external single-cell reference information. So we call each of the center of the hexagon as a grid, as an anchor point. You can imagine we can slide this hexagonal grid and provide a denser lattices of anchor points. So each of them are still a mixture because it contains a rather large area. So the key step in FICTURE is to allow each pixel to look around to pick which anchor point to go to, and update. So the anchor will collect those pixels and update its own factor assignment. And optionally, we could also update the factor level question profile periodically. So the output of this method is a pixel level probabilistic factor or cell type assignment. So to put this intuition into a model, FICTURE is an extension of latent division allocation. So here, similar to LDA, we have a global parameter is over K cell type or latent factors. Each of the factors is associated with average gene expression profile. So this distribution is if you have an external reference panel, you can put the cell type specific expression as this data. Then we have anchor points. The small n is the number of anchor points is often at the scale of millions. So those are the center of the hexagon that we just saw before. Big N here is the number of pixels. So compared to just cell level or anchor or grid level analysis, the difference is for each of the pixel we have an additional hidden variable C here. That's which of the anchor this pixel belongs to. So the nearby pixels will share information through the anchor point, and nearby anchors will share information by the pixel that they share. Because we assume that cellular heterogeneity is really high. So the pixel to anchor assignment is based on a spatial k-nearest neighbor that is so that different anchors and pixels are only coupled locally. So then, based on its anchor assignment, pixel can sample a factor based on the anchors. Sorry, I didn't introduce that. So theta j here is the distribution over k factors of each of the anchor. So each pixel will based on its anchor assignment to pick a factor, and then the gene expression is generated from the factor specific expression profile. Any question on this model? Okay, so next I will first show you a little bit simulation result to show how this method actually works. So in this simulation, we pick 10 cell types from a single cell reference panel, and we assign 8 cell types to different spatial locations. And we sprinkle the other 2 cell types uniformly across the space. So this is to represent the real data cases where you have immune cells infiltrated in the tissue. We simulate 3 different cell types. circle ones, elongated ones, and the more narrow sharp ones. The cell type, cell size are similar to what we see in the real data. So we use this simulation to compare with two representative methods. So FICTURE is the most accurate high-resolution analysis method to our knowledge, and it uses Markov random fields. So it's designed for imaging-based technologies with relatively small field of view. And it's also the biggest limitation is only works for small number of genes. So in this simulation, we downsample the single cell data to only 500 highly variable genes so that FICTURE can perform. Graphite is a more recent method based on the popular graph neural network. So, but it works on a lower resolution that it needs either large grid or segmented cell. So for that purposes, we provide a true cell segmentation for GraphST. So in the result, on the left shows the FICTURE result, and middle is the FICTURE result. So visually, we are pretty similar, achieve similar level of accuracy, and GraphST tend to overcluster. It may be cleaner if we look at the pixel level arrows. So here we only visualize the pixels if the cell type assignment is wrong. On the left, we show the FICTURE's arrow is mostly at the boundary between cell type. That's understandable. FICTURE's arrow is more uniformly distributed, and GraphST just tend to misassign many cells because we're over smoothing. But in reality, we don't really know the proper number of cell types to use. So in this, we also evaluate the cases where we input the wrong number of clusters or cell types. So we simulate with 10 cell types, but we ask each method to cluster into 20 cell types. And for evaluation, we find the top 10 factors or clusters among the 20 that best overlap with the 10 simulated cell types. So any pixel that are assigned to other than the top 10 factor will be classified as error. So here, FICTURE's error pattern does not change much. Whether error rate increase, and GraphST tend to overcluster to reach the defined number of clusters. To quantify this observation, on the x-axis is the targeted number of clusters or factors, and the truth is 10. The y-axis is the pixel level error rate. The blue one is FICTURE, the red one is FICTURE. We also tried another scenario where the cells are more sparse, and the background outside the cells tend to have lower transcript density. And in either situation, the quality pattern of the method are similar. So we perform similarly with FICTURE in terms of accuracy, but the biggest advantage is FICTURE is a lightweight and very scalable method. So here I'm showing the memory requirement for analysis. FICTURE works at a fixed memory budget. FICTURE's memory scale linearly with number of transcripts. And here, even in the largest simulation, it's only 2% of one slide of the real data. GraphST works at cell level, so it's more efficient, more scalable, but still the projected cost for analyzing real data, the memory cost is huge. In terms of runtime, FICTURE is the runtime is linear with number of pixels and is parallelizable. FICTURE runtime appear to be superlinear, and it does not scale to the large number of transcripts. And FICTURE's pixel level analysis is as efficient, as fast as the GraphST cell level analysis. So to summarize observation from the simulation, we achieve comparable or better than the state-of-art method in terms of accuracy. Our FICTURE is vastly more efficient and scalable than existing method, and its turn seems to be robust to over specified number of clusters.
Yichen, we've got a question online.
Yeah.
Okay, yep. So, Chen asked, if cell segmentation is available, can it be used as prior for FICTURE inference?
Well, yes, you could try to use that in different ways. If you believe your cell segmentation is good enough, I guess you can use cell level expression to do your initial clustering and use that as your sector specific expression profile. And if you think, but I'm not sure why. I guess the point is if you have imperfect cell segmentation, can be used to can FICTURE be used to improve that? I guess it's okay. So there are two ways that. So based on our current implementation, I can only think about that you can use your segmentation to initialize FICTURE, but it's possible to extend our method to incorporate some of the cell segmentation information and try to improve that, because in the end, what we got is a pixel level cell assignment that may not be consistent with the initial segment.
It would seem that FICTURE would be more appropriately used as a prior for the segmentation.
Yeah.
Rather than the other way around.
That's what we are currently thinking about. That's right. So I guess if you have nuclear information and cell boundary marker, you can use that as a prior for FICTURE as well. But yeah, thank you.
Yeah, another online question. Oh, yeah, a couple other online questions. So a question from fan said, is the error rate stable among different cell sizes?
Oh, yeah, that's a great question. So we haven't evaluated that, but I would assume, so we haven't evaluated that, but I have some iteration from the real data that, not really the cell size, but how your neighbors are similar with the cell. So if you have bigger cell, that's easier for sure. Even if you have smaller cell, but they tend to be located in homogeneous region, then the problem is still easier because your effective cell size is bigger. But if you have smaller cell that are intermingled with smaller cell from different cell type, or smaller cells in between bigger cell from different cell type, then that's a difficult problem. We acknowledge that. The other question is segmentation 3D? Not yet, but I think it's conceptually easy to extend to 3D. But essentially, what we need is is a prior of the assignment between pixel and anchor, then that's easy to extend to 3D. Yeah, that's all great questions. Okay. So yeah, okay. So next I will show FICTURE in real data examples from these 4 different technologies. So first, here's a 6-scope mouse column example as a motivating example. In this data set, on the top we show the histology image from H&E staining. In this data, we don't have nuclei staining or cell boundary staining, so cell segmentation is not available. At the bottom, we show if we apply a previously proposed grid-based analysis. Here, each of the hexagon size, the side length is 14 micrometer. So we do recover the general structure of the tissue, but most of the details are lost. When we apply to FICTURE, more faithfully recover the fine scale structure of the tissue. We can, it's not shown clearly. So here the red dots in between the clonal pricks are immune cells that are clearly identified. So next we will zoom into this orange box. So on the left is the histology, shows distinct layers of the colon tissue. On the right is FICTURE pixel level assignment. So we see clearly the smooth muscle fibroblast orange, the stem cells and goblet cells are clearly identified, and this green arrow points to the neuronal cells that we see in the smooth muscle layer. For comparison, the low resolution grid-based analysis loses the fine scale tissue structure, and if you use smaller size of the grid, the noise is much more noisy comparing to the instead of adding more details. Okay, the next example is from StereoSeq. It's also a next generation sequencing based technology. So here shows a 16-day mouse embryo section. The tissue area is quite big, it's over 1 square centimeter. And it has nuclei staining, so the original study was done with nuclei-based cell segmentation. And almost all genes are present in this data. And overall, 750 million molecules are captured, but less than half of them are assigned to any cell. So on the left is the published cell level annotation, and on the right is FICTURE result. So each color here shows either the annotated tissue type or our inferred factors. So one interesting difference is in the muscle tissue that we will zoom in next. So during myogenesis in development, myoblasts are matured into myotubes, and then they fuse together to be myofibers. So cell segmentation is particularly difficult in this later stage, where cells become multinucleated and elongated fiber-like cells. On the top is the nuclei staining image, and at the bottom is the cell segmentation-based analysis. So here in this view, we don't see any fiber-like muscle structure, and almost all cells in this region are labeled as myoblast. However, when we apply FICTURE to this data set, we see clearly the heterogeneity within this muscle tissue. So here on the top is FICTURE result with each pixel colored by inferred latent factors. So we see this elongated muscle fibers emerging in this differentiating tissue. At the bottom, we highlight two factors. The red pixels are from myoblast, and the yellow pixels are from the differentiating striated muscle cells. So those factors are labeled because they're distinct macrogenes. So FICTURE also works with imaging-based technology. So this example is 10x linear human breast cancer. The tissue area is about 40 square micrometer. It also only has nuclei staining, and this gene panel contains only 300 genes, and 3/4 of the transcripts are assigned to any cell. We will zoom in this region around. Yeah, so on the top right is the histology image, shows that shows the adipose tissue. So here we have this empty spot because adipocytes have a large lipid droplet where barely any RNAs are present. For adipocytes, the nuclei is tiny and located at the boundary of the adipocyte. And often there are other immune cells located between the big adipose. So on the top left is linear self-segmentation based analysis. So you can see clearly for adipose tissue, the self-segmentation is kind of misleading. And indeed, at the bottom, we show the nuclear staining. The nuclear staining in this region is sparse and does not really recapitulate the histology. So in this case, the top right is FICTURE's result. At the bottom, we highlight two latent factors. The yellow one is adipocytes, the blue ones are endothelial cells. At the left-hand side, we highlighted a couple of immune cell populations. So overall, I think our pixel level analysis provide a more realistic biological view of the underlying tissue. The last example I want to share is from Yichen mouse liver data. So this tissue area is much bigger, over 3 square centimeter. And this data contains both nuclear and cell boundary markers. But still, with this high quality data set, only 3/4 of transcripts are assigned to any cells. We will zoom in this region with near the photo vein. So on the left is FICTURE's influence around the photo vein, and the yellow box highlight bioduct. In the middle, we show the nuclear and boundary staining around the bioduct. You can see that there are small cells tightly packed around the bioduct, and the nuclear staining is not really clear. And the cells lining this photo vein, the structure is unclear. So indeed, cell segmentation is really challenging in this region, and it's basically cell segmentation gave up in this near the photo vein, near the bioduct. So on the left, we highlight several non-parenchymal cell populations as inferred by FICTURE. But although we do not provide cell level segmentation, we can clearly delineate the cell type boundary. Yeah. So I have a little bit more time than I thought. So I will just go ahead to show you another a few supplementary information. So I want to emphasize that cell segmentation is not only efficient in that it captures only small proportion of the transcript, but it's also biased. So here with the stereo-seq mouse embryo data, we show here each of the dotted one gene. On the x-axis, we show the total number of transcripts. On the y-axis, we show the proportion of transcripts assigned to any cells. So we see clearly that not all genes, so not all tissues are equally well segmented. Some of the genes are particularly hard to be segmented into cell, and they have distinct enriched tissues. So if we see how to group the segmentation efficiency by tissue, x-axis is the labeled tissue, y-axis is the proportion of transcript assigned to any cell. The overall average is about 40%. On the left is the tissue that are most poorly segmented. On the right-hand side is the better segmented. Yeah, so similar thing can be said for imaging-based technology. So here in linear breast cancer example, again, we plot the gene by phototranscription, the proportion of unassigned to cell. Is the adipose, this is the microgene for adipose tissue as we saw before. Okay. So in summary, we present a segmentation-free analysis tool that's achieve high resolution that infers cell type at sub-micron resolution for each individual pixel. It's pretty general that it applies to any subcellular level spatial transcriptomic technologies. And it's highly scalable, that's it's orders of magnitude more efficient than existing methods. And it's suitable for emerging large data sets. We want to argue it is less biased compared to the imaging-based segmentation-based analysis, because we see that cell segmentation is often unavailable for sequencing-based platforms, and is incomplete in two dimensions because the nuclei and cells are only partially captured. And it's also biased against multinucleated cells with irregular shapes or sizes, and it's completely ignore the actual cellular analysis. With that, I will thank my advisors and collaborators, and our preprint and code is available. Yeah, thank you.
I was curious how you were gonna do some more of like the downstream approaches that you usually get with segmentation methods, so similar to receptor ligand interactions. Are you able to kind of do this with this segmentation-free approach?
Yeah, that's a good point. For this specific cell communications application, I think it is hard. I will argue that it's better if you have slightly more information about the cell morphology, because what I think we can achieve is, for example, conditional on the factors. See what genes are enriched at the boundary between factors. So we can do that, but I will be cautious that if you want to do cell segmentation, precise cell segment, if you want to do the ligand finding the precise segmentation is more important, is very important, and it's better if you have more information on the cell morphology. But with only our method, I think you can do the conditional analysis of conditional factors to see which genes are enriched at the boundaries between. Other downstream analysis we have considered, for example, the cell type specific spatial spectrum pattern. But again, most of the downstream analysis of cell level, downstream analysis after you do cell segmentation is based on cell type. In cell segmentation, after that, first thing is to do cell type and then do downstream analysis. So you can conditional on pixel level factor the same way as you conditional on the cell level cell type assignment.
Thanks.
Well, first of all, this is just spectacular work, and I'm happy to live in a universe where this is even possible. But my question is, does sub-micron resolution imply subcellular resolution? Or that is, how much inference can be made about the subcellular distribution of molecules from the resolution that you're getting, spatial resolution that you're getting?
Yeah, so I think technically it's possible, because to get, I think two important factors. One is the spatial resolution. The other is how much your transcript diffuse during the whole process. So if that level of diffusion is below the subcellular, it's below the subcellular level spatial pattern, then I think we could do with this data. But the other constraint is how many transcripts you can measure within the cell. I think with the continuous improvement of the technologies, we can certainly do that, but we haven't looked at that yet.
Thank you.
Any other questions? Seeing any in the room? I'm not seeing any online, so let's thank our speaker one more time. Thank you all for coming, and as a reminder, no tools and tech next week, so the next one's on March 7th.